Hello everyone. In this video we are going to discuss about glycolysis or the glycolytic pathway. Glycolysis was the first metabolic pathway to be elucidated. The word glycolysis is derived from two Greek words, glykis, that means sweet, and lysis means splitting. So basically, a molecule of glucose is degraded in a series of enzyme catalyzed reactions to yield two molecules of a 3-carbon compound known as pyruvate. And during the sequential reactions of glycolysis, some of the free energy that is released from glucose is conserved in the form of ATP and NADPH. Next, before we proceed to the steps of glycolysis, it is essential for us to know the importance of glycolysis. So firstly, glycolysis is an almost universal central pathway of glucose catabolism and the pathway with the largest flask of carbon in most cells. Secondly, the glycolytic breakdown of glucose is the sole source of metabolic energy in some mammalian tissues and cell types. For example, erythrocytes, renal medulla, brain, and sperm. Thirdly, some plant tissues that are modified to store starch. For an example, potato tubers, and some aquatic plants, for an example, watercress, derive most of their energy from glycolysis. Also, many anaerobic microorganisms are entirely dependent on glycolysis. Also, the intermediates obtained from glycolysis act as a major source for the synthesis of many other compounds. For example, 2,3-bisphosphoglycerate is used for the release of oxygen in RBC. Dihydroxyacetone phosphate obtained from glycolysis is used in triacylglycerol synthesis, and pyruvate is used for alanine synthesis. Next, we would look into the location and types of glycolysis. So glycolysis occurs in the cytosol of the cell. It is basically of two types, aerobic glycolysis and anaerobic glycolysis. Aerobic glycolysis takes place in the presence of oxygen, yielding pyruvate along with the production of 8 ATP molecules. And this aerobic glycolysis is the pathway that provides energy for all the tissues, wherein brain alone derives 80% of the energy from aerobic glycolysis in fasting state. Whereas anaerobic glycolysis takes place in the absence of oxygen and it yields lactate along with the production of 2 ATP molecules, and anaerobic glycolysis is the source of energy for erythrocyte and lens. Also, the vigorously contracting muscles derive its energy from anaerobic glycolysis. Moving on to the steps of glycolysis. Glycolysis is an extra mitochondrial pathway and is carried by a group of 11 enzymes. Glucose is converted to pyruvate in a 10-step reaction. Out of these 10 steps, 3 steps are irreversible while the rest are essentially reversible steps. The glycolytic pathway can be divided into 3 major phases: preparatory phase, splitting phase, and payoff phase. Preparatory phase or glucose activation phase or energy investment phase basically traps the glucose and prepares it to move into the next phase. Here, 2 molecules of ATP are invested and the hexose chain is cleaved into fructose 1,6-bisphosphate. steps 1, 2, 3, 4 together constitute the preparatory phase. Splitting phase basically cleaves the fructose 1,6-bisphosphate into two triose phosphates, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Next, the payoff phase or the energy generation phase or the energy extraction phase basically converts the triose phosphate, that is glyceraldehyde 3-phosphate, to pyruvate coupled with the formation of ATP. Next, we begin with step 1, uptake and phosphorylation of glucose. So here a 6-carbon glucose is phosphorylated to form glucose 6-phosphate catalyzed by a specific enzyme called hexokinase. So phosphorylation basically refers to the addition of phosphate using ATP. Now the enzyme hexokinase can be related into hexo and kinase. That is, hexo refers to or relates to the 6th number of carbon present in glucose, and kinase refers to the phosphorylation using ATP. So the H plus ion from carbon number 6 in glucose is released, thereby making the reaction an exergonic reaction, and a phosphate molecule from ATP is added to carbon number 6 of glucose, making glucose into glucose 6-phosphate. Here the glucose becomes more stable in the form of glucose 6-phosphate, thereby gaining more reactivity so that it can proceed to the next stage, that is splitting phase. Step 2, isomerization of glucose 6-phosphate to fructose 6-phosphate. This stage is basically a isomerization reaction, that is an aldose-ketose isomerization that takes place spontaneously. The prime aim of this step is to have a symmetry in the molecule so that two identical 3-carbon molecules can be produced in the next phase, that is splitting phase. So glucose 6-phosphate is isomerized to a symmetrical molecule, that is fructose 6-phosphate, by the enzyme phosphohexose isomerase. Here there is an opening of the glucopyranose ring of glucose 6-phosphate into a linear structure, which later changes to the furanose ring structure of fructose 6-phosphate. Step 3, phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate. This highly spontaneous reaction has a mechanism similar to that of hexokinase. The prime concept in this step is stabilizing the molecule, making it more reactive, more energetic. So here phosphofructokinase 1 transfers a phosphate group from ATP to carbon number 1 of fructose 6-phosphate, converting it into a more energetic fructose 1,6-bisphosphate. This reaction is irreversible and 1 ATP is used for phosphorylation. Step 4, cleavage of fructose 1,6-bisphosphate. This step marks the beginning of splitting phase. Here the 6-carbon fructose 1,6-bisphosphate is cleaved into two 3-carbon units, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The enzyme that catalyzes the reaction is aldolase, and therefore the mechanism of the reaction is an aldol cleavage reaction. Step 5, interconversion of the triose phosphates. Glyceraldehyde 3-phosphate is on the direct pathway of glycolysis, whereas dihydroxyacetone phosphate is not. Hence, triose phosphate isomerase converts dihydroxyacetone phosphate into its isomer glyceraldehyde 3-phosphate for generating ATP. Thus, the net result is that glucose is now cleaved into 2 molecules of glyceraldehyde 3-phosphate that further proceeds into the next stage, that is energy generation phase. Step 6. Oxidative phosphorylation of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. This is the first step in the payoff phase. Here, the glyceraldehyde 3-phosphate is oxidized to 1,3-bisphosphoglycerate and the reaction is catalyzed by glyceraldehyde 3-phosphate dehydrogenase. It is the energy yielding reaction. Reactions of this type in which an aldehyde group is oxidized to an acid are accompanied by liberation of large amount of potentially useful energy. During this reaction, the enzyme dehydrogenase transfers the hydride group and NAD+ is reduced to NADH. Step 7. Conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate. The enzyme phosphoglycerate kinase transfers the high-energy phosphoryl group from the carboxyl group of 1,3-bisphosphoglycerate to ADP, forming ATP and 3-phosphoglycerate. Now, this is a unique example where ATP can be produced at the substrate level without participating in electron transport chain. This type of reaction where ATP is formed at substrate level is called as substrate level phosphorylation. Step 8. Conversion of 3-phosphoglycerate to 2-phosphoglycerate. Here, the enzyme involved is phosphoglucomutase. Mutase shifts the phosphate group from 3rd carbon to 2nd carbon, thereby converting it into 2-phosphoglycerate. Step 9. Dehydration of 2-phosphoglycerate to phosphoenol pyruvate. This reversible reaction involves the conversion of 2-phosphoglycerate to phosphoenol pyruvate by the enzyme enolase. One water molecule is removed and a high energy phosphate bond is formed. Step 10. Conversion of phosphoenol pyruvate to pyruvate. Phosphoenol pyruvate is dephosphorylated to pyruvate by pyruvate kinase. First, phosphoenol pyruvate is made into a transient intermediary state of enol pyruvate, which is spontaneously isomerized into ketopyruvate, that is the stable form of pyruvate. Here, one molecule of ATP is generated during the reaction, and this is yet an example of substrate level phosphorylation. Next, on a quick note, let us revise the key points involved in glycolysis. Glycolysis involves the conversion of a molecule of glucose into 2 molecules of pyruvate. It occurs both in aerobic as well as anaerobic condition. There are 3 stages in glycolysis: preparatory phase, splitting phase, and payoff phase. It is a 10-step reaction where 3 are irreversible and the rest are reversible. Step 1, 3 and 10 are irreversible steps, whereas all the other steps are reversible. Preparatory phase involves the investment of 2 ATP molecules, whereas in energy generation phase, 2 molecules of NADH and 4 molecules of ATP are produced. Lastly, moving on to the energetics of glycolysis. So the energy yield in aerobic glycolysis is 8 ATPs. So here we can look at the energy generation steps. So hexokinase involves the investment of one ATP, phosphofructokinase involves the investment of another ATP, glyceraldehyde 3-phosphate generates 2 molecules of NADH, that is equivalent to 6 ATP molecules. Phosphoglycerate kinase and pyruvate kinase contributes 2 molecules each of ATP using substrate level phosphorylation. So altogether we can see there are 10 ATP molecules. But since in preparatory phase there is already an investment of 2 ATP molecules, so we would deduct 2 ATP molecules from the total of 10. So this implies to 8 ATP molecules generated in aerobic glycolysis. Next, in case of anaerobic glycolysis, hexokinase employs the investment of one ATP, phosphofructokinase involves another investment of ATP, whereas phosphoglycerate kinase and pyruvate kinase contributes each of 2 ATP molecules using substrate level phosphorylation. So altogether there is a total of 6 ATP molecules, and since 2 molecules are already invested in preparatory phase, so a total of 2 ATPs are finally produced in anaerobic glycolysis. I hope this video helps you in understanding of glycolysis, and thank you for watching the video.